All right. Well, good afternoon, everyone. I see the line has dwindled down, so I think we're going to go ahead and get started. We'll probably run a few minutes late, but I think that'll be all right. Before we get started, I wanted to recognize the land that we're standing on. Augsburg's spirit is propelled by a heartbeat that started long before us. That heartbeat was present before our founding, when the land where Augsburg stands today was stewarded by the Dakota people. The Dakota are the original inhabitants of this area, and they are still here today. We honor their wisdom about this place, their recognition that we are all a part of the same creation. We share their sense of obligation to the larger community, including to future generations. So thank you. Thank you all for being here today. Before I get started with introducing our speaker today, I wanted to say a few thanks. First of all, I want to thank the Sverdrup family and the Minnesota NASA Space Consortium, I think I said that right, for funding and making this event possible. I also wanted to thank, I'm not sure if she's here, Shannon, Obey, and Augsburg Events for helping plan all of this, and also TJ in our tech support. As you're about to see, we have lots of really cool videos and audio to show you, so setting that up was really critical. I also wanted to thank all the faculty for advertising these talks, and also to the students for coming today. Okay, so I'm going to do my best to keep... Oh, yes, okay. So it's basic. I'm going to go as quickly as possible. But today we are so fortunate to have Dr. Janet Iwasa with us, a molecular animator who's from the University of Utah, whose work inspires so many across literally planet Earth to see invisible worlds of molecular biology. I still remember as an undergraduate the first time I saw molecular animation. At that point in time, YouTube wasn't really a thing yet beyond like viral sensations like Charlie bit my finger and Leave Britney alone. So there wasn't really a way to know that like these animations existed. And I was sitting in this molecular biology class, and we'd been talking about gene expression and the first step in gene expression. I don't want to get too technical, but it's called transcription, and it involves this molecular machine called RNA polymerase. Okay? When the movie started playing, and I'm not over-exaggerating, I almost fell out of my chair. I remember looking around at my peers, and I'm like, is no one else seeing what I'm seeing? Like, excuse me, like, what's happening right now? Students, I invited Dr. Iwasa here today to be the Sverdrup speaker, not only because she's a world-renowned molecular animator whose work has scientific merit, but because she represents a part of my own undergraduate experience where I found hope in these animations, which might sound weird to you, but I did. When something deep down clicked inside of me and I was changed forever. As your instructor, I do my best to show you this, to awaken some deep epiphany inside of you. I can't describe it, but when you experience it, you know what I'm talking about. Even if at the end of the talk today, if you still don't see yourself pursuing a career as a molecular animator or even as a scientist, that's okay. I want you to simply know that simply surrounding yourself with people who are on fire about what they do is critical. It will help you find whatever that is that you want to do, even if it's seemingly not related. Real quick, back to molecular biology. So after the class was over, I almost tackled my instructor, and I had so many questions for her. How does this RNA polymerase move so quickly without making mistakes? What was that weird DNA bending thing that happened? And it seemed to like do like a molecular kiss, and then RNA polymerase is off to the races. And most importantly, why did you wait until my 4th year of college to show me this? Can you please just show that to me right away? Like, she was a little scared, and so she gave me her own personal copy of the DVD. I still have it here today. It says DNA Interactive. It's a little scratched up. I checked it last night. It still works. You can see I didn't return it, unfortunately. But when I went back to my apartment, sorry, I'm going a little long. I'll wrap it up. I watched it for like 2 or 3 hours, these animations. And then something peculiar started to happen. Like I thought initially it was my neighbor playing some music, but it wasn't. Like, I could hear actually music, okay? I am so dead at, serious. Like, this thing, what I'm trying to say is this thing, this RNA polymerase, it was dancing, okay? So, I wrote like way too much here, so I'm gonna cut it short and just say that Dr. Iwasa, if you ever want to collaborate and open like a dance club, I've set music to all of these animations, and in private I dance to it all the time. We could, it would be fun. Maybe help fund some research. Students, even if you're here today primarily for extra credit, I hope that when I see you in classes, I see you're worried and you're stressed out. And I think you're craving something deeper. I hope that's what you start to feel today. It'll feel a little bit like indigestion, but it's actually, there's something moving inside of you that's saying, it's more about the grade. I don't know, I want to be on fire. That sounds terrible. Okay. Like, I don't know. So, it reminds me, that's what I think is amazing about these animations. It brings out so many thoughts, feelings, emotions in different viewers. It shows that scientists aren't all the same. Some are sensing and other are intuiting. And it reminds me of the quote that the great author, poet, activist Maya Angelou once said, "Everything in the universe has rhythm. Everything dances." All right, so I'm gonna do it. It's not really, all right, everyone, it's about that time, okay? Please join me, Dr. DDDJJJ, molecular. I don't know, in welcoming Dr. Janet Iwasa to Augsburg.
I love having that soundtrack to me walking on stage. It's so great. Thank you so much to Dr. Tan for the invitation to come here, and also for just being such an incredible host, and thank you for that introduction. So I'm going to talk to you today about what I do. And so when people ask me what I do, I often say I'm a molecular animator. And that often begs the next question, like, what is that? I'm like, well, I like use software like the software that Pixar uses, but instead of animating like fish, I'm animating molecules. And you know, I kind of, the way I think about this is this kind of work follows a very, very long tradition that harkens back to the ancient days of illustration. And so, you know, scientific illustration is quite an old field. One could argue that this cave painting in France is an example of a scientific illustration where, you know, the illustrator is thought to have been showing where on this horse-like creature you want to point that arrow at in order to get, you know, kind of a fast kill there. So this is considered kind of an old illustration. And Leonardo da Vinci, the epitome of the term Renaissance man, was not just a scientist, he was an artist and engineer, and he was an inspiration to many scientific illustrators who also bridge the field of art and science. And so for many scientists throughout the ages, many of the ones that we know today are famous partially because they were gifted at their observation skills, but also their illustration skills. They were able to kind of show the world the things that they saw. In this case, you are seeing the invisible to some degree, right? So the kind of the inner workings of the human body are not things that are readily available, visible to people, but Da Vinci made it possible for others to see that. Galileo kind of went in the other direction, right? So using a telescope, he was able to observe the moon. And, you know, for the first time, he was able to see that the moon wasn't this perfectly smooth sphere, which is what everybody thought it was, but it was actually all craggy and bumpy. This was surprising to a lot of people. So a lot of the observations he made were considered extremely controversial. And so his observation of Venus and the phases of Venus made him realize that Earth was not the center of our solar system, but the sun was, which led him to have lots of problems in his life. But, you know, like, he is also an example of a scientist who was able to create these beautiful illustrations of the things that he observed. On the microscopic scale, Robert Hooke is credited for he created this kind of a microscope using like a tiny glass bead that was kind of embedded in a kind of a plate, and he would just hold it up to his eye and take a look at things. So this was a microscope of his own design. And he would create illustrations like this. So this book called Micrographia has all of these beautiful illustrations, kind of really huge, larger than life. Like this is a flea, and people would take these illustrations. So there aren't that many books of his that are kind of intact, because a lot of people rip these pages out and then put them on their wall. They were considered art. The flea, I'm not sure. Maybe that's why it's still in here. Maybe people weren't that into putting a flea on their wall. But he's also credited for coining the word cell. And this is an illustration of cork that he took based on his observations. So again, another person, a scientist who has, this kind of the gift of being able to observe and to kind of create those drawings that can really show everybody else what he's observing. Moving kind of closer into the current age, Santiago Ramon y Cajal is considered the father of modern neurobiology. And so prior to Ramon y Cajal, people thought the brain, they didn't realize it was made up of all of these different cells that were touching, that were communicating with each other, which is, of course, what we know today to be neurons. So Ramon y Cajal basically observed these slices of brain and drew out these beautiful illustrations of different cells, of different neurons. And it kind of basically made him realize that these aren't just interconnected cells, they're these touching cells rather than a continuous network, which is kind of what people thought they were before that. So I think, this is to show that a lot of the famous scientists who are really credited with starting new fields were able to create these beautiful illustrations of the things that they observed, and this helped them make a lot of their discoveries as well. So nowadays, of course, we can use things like microscopes in order to create these images. So this is a Purkinje cell, the same kind of cell that Santiago Ramon y Cajal is illustrating here. And so, using things like microscopes, we no longer have to really be great artists. And so that's, I think, led to sort of the decrease in the number of illustrator scientists, right? We have all of these great tools. We don't need to just kind of sit down and draw everything. But I would argue that is not really the case when it comes to molecular level things, where we still have to bring back our ability to illustrate, and in my case, animate, in order to kind of show what we understand. And why is that? So the main reason for why we need to have things like molecular animation and illustration is that molecules are tiny. So this is an interactive web-based visualization that I encourage you to go check out. It's at learn.genetics.utah.edu, which is a project I'll be talking about later. But using this, you can basically zoom in, starting with a coffee bean. You can zoom in smaller and smaller until you can start seeing things like viruses, as well as molecules. And so this kind of linear scale really depicts how small molecules are. And so this is why they're so difficult to be able to study and to be able to understand. So molecules are smaller than the wavelength of light, which means that we can never use any sort of light-based microscopy to observe them directly. We will never be able to just use a light microscope to look inside a cell and see molecules. That is not possible. Instead, we have to use other types of techniques like X-ray crystallography, like using basically all of these other kind of wavelengths of light to be able to observe molecules. And oftentimes, we need to use techniques that basically will have to freeze things. So we can't see moving molecules within the context of cells using really any technique that we have today. So this makes, kind of basically understanding the world of molecules quite difficult. So what do scientists do in order to kind of describe what molecules do? So we have a lot of different ways of collecting data. And so we have a lot of indirect ways of figuring out how big proteins are, where they might be in the cell using things like microscopy. So using a light microscope, we can basically localize where molecules are, maybe how they move around as well. And using other techniques, such as X-ray crystallography and electron microscopy, we can figure out the size and shape of proteins through a lot of different averaging kind of techniques. So all of these different types of experiments that are often indirect will allow us to start creating this sort of an illustration, a drawing of how we think molecules work inside cells. So these are some of the illustrations that I got from my collaborators. So scientists often need to kind of draw in order to explain things. So more often than not, when I talk to a scientist about their work, they're like scribbling madly on a piece of paper while they're talking. And so that's usually the kind of the basis of a lot of our animations that we create. I always take these illustrations and refer to them. So, at the end here, what we're talking about is that visualization in science, in molecular biology in particular, is not about recording our observations, because we have great ways to do that using technology, but it's about being able to visualize our deductions. From all of these different observations that we're able to make, what do we conclude about what's happening inside the cell? So I just created this illustration this morning using Google AI, and I was like, give me an illustration of a woman thinking about proteins. And I don't know, like I guess those little blobs look kind of like proteins, but I was like, it's kind of cute. So I used that, but some of the proteins, they actually kind of look sort of realistic. So, this led me in graduate school to this idea of basically why do we need animation in biology? And this is because we can basically use these animations, the same animation software that places like Pixar use, to create a visual hypothesis of how we think things work from all of this experimental data. And so I've been really lucky that, this is something that was not a field really. And so I've been very fortunate at the University of Utah to be able to start my own lab that focuses on molecular animation and visualization. The top 4 people here are people who are currently in the lab, and the bottom 8 are people who have been in the lab prior. And so, over the years, we've created a lot of different animations of lots and lots and lots of different molecular processes. These are largely in collaboration with scientists around the world. Basically working together with them has been a true joy. So really, kind of a lot of these animations are really the result of many, many years, sometimes decades worth of research that is taken to be able to kind of show on the molecular scale what's happening. So I'll show you a couple examples of these projects. One of the longest running projects in my lab is called The Science of HIV. This is one that I started in 2014, soon after arriving at the University of Utah. So this project was really started as part of a large grant-funded group, NIH-funded group, that's called the Cheetah Center. This is based out of the University of Utah. It includes 20 researchers from around the country who are all interested in understanding the structural biology of HIV. And so when I first arrived at the University of Utah, I proposed this idea that I wanted to create an animation of the life cycle of HIV. And the reasoning for me to think about doing this was that I thought about the fact that we have spent probably billions of dollars on HIV research over the years. And as a result of that research, we understand a lot about how HIV works. But if you were to talk to a member of the public about HIV, I think what most people can tell you is that HIV causes AIDS and that there's no cure. And both of those things are still true. So I think, the problem here is that it doesn't really describe all of the things that we know about HIV. We understand why it's been so hard to cure. And we also, basically have so much knowledge that I can make, molecular level animation of the entire life cycle, almost to completion. So I'll show you part of that animation now. And it has some sound.
HIV first enters the body through the bloodstream. The viral particle is surrounded by a membrane that's studded with viral proteins known as envelope proteins. In this cross-section, the conical-shaped capsid, shown in orange, houses the RNA genome, shown in blue. Accessory proteins are shown in purple. At about 100 nanometers in diameter, HIV is about 100 times smaller than its target cell, the T cell, shown here to scale. The virus first contacts T cells through interactions between the viral envelope proteins and membrane proteins on the T cell surface. Initial binding occurs through CD4 proteins, shown here in yellow. Binding of a second membrane protein, called a coreceptor, shown here in orange, triggers a dramatic change in shape of envelope protein. The first conformational change allows envelope protein to insert into the membrane of the T cell. Next, envelope protein folds back on itself, forcing the fusion of the T cell membrane with the viral membrane. This allows the viral capsid to enter the cell.
So you can watch this animation online. So I'm not going to play through the whole thing because it's like 8 minutes long. But what happens is that the capsid moves along what's thought to be microtubules while it's moving towards the nucleus of the cell. And during that time, the viral genome, which is an RNA genome, gets copied into double-stranded DNA by a protein known as reverse transcriptase. By the time it gets to the nucleus, you have a full copy of the viral genome in DNA form, which then can integrate into the host genome. This virus is known as a retrovirus, so it's able to integrate its genome, its DNA-based genome, into the cell. And basically, this is the major problem that has been hard to kind of understand, is how we can find these cells with these integrated genomes within them. So these genomes, these viral genomes, can live inside cells for days, weeks, decades without being activated. But once they do become activated through a process that's still kind of poorly understood, it can start basically creating these genes. These viral genes that are being shown here, and this mRNA from the viral genome can get exported out of the cell. Some of it goes to ribosomes, which get made into viral proteins, and some of them get packaged into new viruses, which is shown here. So this is the budding stage, where you have basically the cell has turned into this viral factory, and now you're creating new viruses from this cell. So it's packaging all of these viral proteins in it. And then there are proteins shown at the bottom here that basically cause fission between what's essentially a viral kind of vesicle and the cell. So that stage is shown here. And then the last stage is called maturation. So in order to get into that viral genome packaged into the capsid, you have a bunch of different events that are caused by protease that allows the formation of this capsid around that RNA genome. And now you have a mature virus which can go on to infect other cells.
Mature viruses are
okay. So this animation is available on this website that we call The Science of HIV. This is a project that has been ongoing for over 10 years. And so we're constantly updating this website. So some of the latest animations we have 2D animations that show how reverse transcription works. This was actually created by an undergraduate summer student in our lab. And we also have just kind of other animations that show how the cell can fight back. So different proteins that are involved in sensing viruses like HIV, and how basically our immune system, in some cases, is able to detect HIV and fight back. We also have animations that show drug treatments. So Science's discovery, or I think breakthrough of the year for 2024, was a molecule known as lenacapavir. This molecule is a very, very effective inhibitor of HIV. We have an animation that shows basically how lenacapavir works to pause certain segments of the life cycle. It makes the capsid fall apart more easily, and then the cell is able to detect it. And so this is a really long-lasting drug, and is considered really is considered a breakthrough for HIV treatment. One of the other things that, I thought about for the Science of HIV project is thinking about how to visualize some of the processes that are really hard to especially show at the molecular level. In this case, I teamed up with TED Ed. So I don't know if, hopefully you've seen some TED Ed videos. They make videos on pretty much every kind of subject. But one of the great things about TED Ed is that they're always open to new ideas. And so basically I pitched this idea. I said, I want to make a video about why HIV/AIDS has been so hard to cure. And I came up with I worked with the writers on the TED Ed team to create this kind of a script, and then they basically hired 2D animators to animate that script. And then they created this video, they put it online, it gets 2 million views, and they were like, we would like to give you a gift certificate for working together with our team. So this is really amazing resource, and I really encourage anybody to, if you have an idea for a TED Ed subject, to think about working with them, because it is really fun and it's a great way to kind of get a lot of different ideas out there. The animation itself is one of the goals was to be able to use it for dissemination to kind of show what all these scientists have been working on and how much we understand about HIV. And one of the great things is that it has been used in lots of different ways. So it's been used in different types of publications and online. And I make these animations available for anyone to use for any purpose. Another major part of this project is also thinking about animations for the scientific community. So the Science of HIV website is really largely kind of meant for kind of a more public audience, but we also create animations for scientists as well within this HIV research group that's really meant to communicate science within the kind of formats that scientists normally communicate, which is to say publications and talks. And this is an animation, this is a collaboration with one of our researchers in the Cheetah Center, and it kind of depicts why the limitations of current scientific software. So this little animation here, so basically the group of Adam Frost and his postdoc Henry Nguyen, they solved a structure of this protein that's known as escort-3, which plays a role in the budding part. So basically when you have a virus that's budding off and it needs to basically break off from the rest of the cell, escorts come in and they basically bind to the membrane and they cause that break to happen. So they had these two different structures, kind of a more open ring and a more kind of confined ring. And this is basically an animation that they could create using the scientific software. So that's kind of shows a bit of the limitations of what they can do. They can basically create this morph between those two structures, but that really doesn't tell the whole story. So what Adam wanted to show was that you can bind the membrane and then you can go from this larger ring to a smaller ring when a second protein binds. And that's really difficult for them to show using that existing scientific software. So I collaborated with them to show this animation where you have the second protein binding, and only after the second blue protein binds do you have this slow constriction of this ring that also kind of basically constricts that membrane and gets it closer to be able to get to that point of fission where that new virus can break off. So, and you can also show the membrane in here. So a lot of kind of information, you can give it some context. And this was published in a paper. So a lot of the animations nowadays are published in papers. It's really becoming part of the way that scientists are able to communicate. Another example of basically actually the same protein and creating animations of that was this other example where I collaborated with researchers also studying escort-3. And in this particular case, they wanted to show that there are different hypotheses about how this constriction happens. And so we made basically 3 different animations showing 3 different hypotheses that kind of look pretty similar if you aren't too familiar with them. But for the researchers, these really have different kinds of predictions of how things form and basically how these proteins are able to constrict the membrane. All right, so I wanted to tell you a little bit about our process. How do we create these animations? So the first step, and this is pretty much analogous to any sort of animation studio like the ones at Pixar or really any animation studio around the world, is storyboarding. So actually the very first step before this is that we have a lot of conversations with researchers about what they're doing and, what are the main players, what are the proteins that are being shown here, and what are they doing? And after that point, we will draw a storyboard. And for me, this is often like a pretty simple sketch, a two-dimensional like pencil and paper sketch of the process and some notes about basically what's happening. So this is an example of a storyboard that I created of HIV. And in this case, I have a collaborator who basically took my storyboard and commented on it. So you can see my notes are in black, his notes are in red. So where he's kind of saying, well, there's a little bit of additional stuff in here, or, this I would change a little bit. And so this is basically a back and forth process between us as the animators and our collaborator, who are researchers often working at the bench and have all of this kind of internalized knowledge about how these things work. And so based on this, we can go back. We often reiterate. We will redraw the storyboard if there are a lot of changes. But otherwise, we would start moving forward with that animation process. And so there are a lot of resources for structural biology. So if you're interested in looking at molecular structures, one of the great resources is called the Protein Data Bank. So for researchers to basically publish a paper about a structure, it needs to be deposited in the PDB. And what the PDB is, if you go there, is you can basically look at a PDB file, a Protein Data Bank file, and it will tell you every basically XYZ coordinate of every atom in a structure. And so you can have all of these beautiful looking structures. You have things as big as viruses, also single proteins, mRNAs, DNA is all kind of in the PDB. We also have AlphaFold. So AlphaFold was a Nobel winning kind of innovation last year, where you basically, based on all of these structures that have now been published in the Protein Data Bank, we're able to now we're able to figure out the structure of proteins that haven't yet been experimentally solved. And so using AlphaFold, we can now predict the structures of proteins and also protein complexes. So these are both things that my lab uses a ton, both PDB and AlphaFold, to create some of our animations. So once we have, this structure of a protein, so most of our animations are molecular, we need to basically put it into a format that we can then import into our 3D animation software. For most 3D animation software, this means making it into what's considered a polygonal mesh. So we can't just import a bunch of atom coordinates into the animation software. The animation software we use is from Hollywood. And so what it's kind of expecting is sort of a polygonal mesh that maybe is a fish or a dog or whatever, but in our case we have proteins. And so what we do is we use scientific software to basically create a mesh out of this protein structure. In this case, we have a protein known as actin that I'm creating this mesh out of that we can then export out of the scientific software, which is called Chimera, and then import that into 3D animation software. We use Maya. So we actually not only use these kind of molecular structures, it's also possible to use light microscopy to also create these structures of cells. So in this case, a 3D light microscopy technique was used to create this movie, this 3D mesh of this cell that's crawling that we then integrated into a longer animation. So this is a view of our 3D animation software. We use a software called Autodesk Maya, but there are several out there that are basically very similar. Blender is one that's open source and free that we've been using a bit more lately. So in this example, what you can see is I took that actin, this kind of a polymer, what's considered a filament of actin, which is made up of a lot of individual proteins, and we're animating basically this kind of process of this polymer forming. And there's different ways of doing this kind of animation. The most traditional way is called keyframed animation, where you basically move the object, keyframe it, move it, keyframe it at different points, and then you can basically animate it moving. That is a pretty long and time-consuming process unless you have just 2 or 3 things. This This is traditionally how most animation is done in the major movie studios. But for us, we usually, because we have a lot of molecules, sometimes hundreds, thousands of molecules, we also use a lot of kind of scripting-based techniques. That I think I show a little bit later on. So there's different ways of like deforming things that you can see here. So in some cases, we're able to use the kind of built-in tools that we have inside Maya. And in other cases, we kind of have to make use of tools that kind of are used for different things. So like in some cases, we have to kind of be clever about what's available and how we use it. So for example, in some cases, if we wanted to make a flexible DNA strand, I would use the tools that would normally be used for creating like hair. So it's called the hair tool. And basically it's for like hair blowing in the wind like this. But you can use it basically to also animate things like DNA and RNA. And so we often have to kind of think about, how do we use these tools that are really made for animating like superheroes and fish and stuff, but for animating molecules and for animating molecular motion. So that's kind of both the fun, but also the challenge of using these tools that were really not made for animating things like molecules, but for animating these molecular processes. So the final step is called compositing. So this is basically the point at which we have, created everything in our animation software. We've exported it as a bunch of image files. So typically, we have 30 frames a second in most animated movies. That means that you export 30 frames for 1 second of animation. And so the typical animations we make are about a minute long, so this is thousands of frames. We can have different layers, so often we'll have like a foreground layer and a midground layer and a background layer. We can all animate those all separately. We can do different effects, like we're blurring things out in the background here so they're not as distracting. We can kind of pump up color saturation. We can add titling, sound, all of that can be done. This compositing software is called After Effects. It's made by Adobe. And it also can be used for basically doing 2D animation. So you can do some animation straight in After Effects, but we tend to do all of our animation in the 3D tool and then import it into our compositing software. So that's just a little bit about our process. A lot of it is pretty much traditional animation. It's really kind of the first part of it where we're really talking to scientists, understanding what they do. That is where a lot of the kind of interesting kind of thoughts and discoveries can happen. And this last part is really kind of more of a traditional animation pipeline. So we have been involved in a lot of different projects. So not only have we been involved in projects that have to do with kind of in research, publishing research papers, and projects like HIV, which is largely a research project, but I've had the real kind of fun of being able to create exhibits at museums as well. And so this is an example of a very recently launched exhibit at the Natural History Museum of Utah. So this is part of the University of Utah, where we created this exhibit about the ribosome. And this was really exciting for us because if you think about it, if you've been to many science museums, there are very few things about anything molecular, like maybe there'll be a structure of DNA floating around somewhere, but that's about it. There's nothing really kind of really showing more stuff than that. But the molecular world is super exciting. And so we were very happy to be able to create this exhibit. And so what this includes is basically the animation that was playing at the beginning of this talk is this animation that's projected in the back of this screen. There's also a touch screen kind of monitor that you can't really see here. It's kind of off in the corner, but it also has this kind of ribosome structure that you can see in the middle. And the idea is that you want to be able to appeal to people of all ages. And so kids basically can take an mRNA that's like kind of rubbery and you can shove it inside this ribosome and turn this little wheel. And then all of these little peptides pop out as like lit up balls out of the ribosome. So it's actually really fun to use, and it's always super crowded. Like kids are always at the ribosome exhibit, which has been, super gratifying to see. So this is the animation that plays in the background. It basically shows the inside of a mammalian type cell. So this type of view is something that you might see if you were able to zoom into your own cells. You can see the ribosome at work here making proteins, reading this mRNA and creating a new protein. And it's basically a very nice example of a molecular machine. We also created, on the touchscreen monitor, you can see an example of the bacterial ribosome. So this animation basically shows the process by which you have these tRNAs that are being brought into the ribosome and forming this new protein strand. So this is shown in real time. So at the museum exhibit, you can push a button to say, I want to see the ribosome in real time, which is showing it this fast, or I want to see it slowed down so you can really see all of those things coming in and out. You can also push a button to basically say, I want to add an antibiotic. So bacterial ribosomes can be, so there's been a number of antibiotics that are used that are basically impacting the bacterial ribosome specifically. So you take these antibiotics, they gum up the works, as shown here. The bacterial ribosome can't work anymore, and the bacterial cell dies. This is specific for the bacterial ribosome. Of course, you have ribosomes in your own cells that these antibiotics can't bind to. And so it's kind of a nice way of showing how different drugs work within the context of this exhibit. We've also been interested in a lot of other educational kind of things lately. So one of the things I started this year is writing these articles that are called the Molecule of the Month. So this is actually part of the PDB, the Protein Data Bank. And I took this job from David Goodsell, who is a really famous, renowned illustrator of molecules, and it was a real joy. So he retired earlier this year, and I took this over for him. And so you can basically go to the PDB, PDB 101, and take a look at all of these articles. So we feature a lot of articles about structures, and you can read about them there. So I encourage you to do that. We're also incorporating animations as well. Another thing that I took up recently, so last year I became the director of the Genetic Science Learning Center at the University of Utah. This is a really great resource. So it's learn.genetics.utah.edu. So there's just kind of a bunch of games and interactives for all sorts of biology. So a couple examples, we have a game about pigeon genetics called Pigeonetics. We also have a ton of animations and videos and interactives. These are used by millions of people in schools around the world. So, you know, one of the interesting things that I did when I was at home, kind of looking at some of the Learn Genetics modules, I was playing with a game that was about dog breeding. It was dog genetics. And my kids looked over my shoulder and they're like, we did that in school, you know, so they just, it's used by classrooms around the world. And so, yeah, it's a great resource. I encourage you to take a look at it. It's also a great team. So this is, Louisa Stark is the former director of the Genetic Science Learning Center. She's been running it for over 20 years. And there's a group of 16 people who are artists, illustrators, teachers, web developers, research and evaluation team that are all part of this group. So it's been really fun to be part of this group and to really be thinking about formal education and not just informal education. And Learn.genetics is not just about genetics. So we have all of these modules. We also collaborate with scientists around the country and have created modules on like geology, on neurobiology, as well as on plants. So I encourage you to take a look. And also, if any of you are interested in collaborating, we're always happy to take on new projects. Another kind of fun kind of thing that we've done as a group is we've tried to teach scientists how to create outreach videos, encouraging scientists to basically make their research more accessible. And we do this through the process of basically teaching them how to do stop motion animation. So we do this within our research groups. So our Cheetah Center, our HIV Structural Biology Center, all the labs in there have created stop motion animations that describes what their lab does. All of the labs in my department have also participated in these workshops and created these stop motion animations and videos about their research. And we also go to local schools and teach kids about basically how to do stop motion and encourage them to create scientific animations. So this one is one that students made about coronavirus and antibodies and how that works. These are like very short workshops where we basically have 45 minutes with a group of kids and we get them to come up with a storyboard, create an animation from start to finish, and then we eat popcorn at the end and watch all the videos. So it's like a very fast process that the kids have fun with, and they're just showing kind of what they've learned in classrooms. So, I just want to show some more animations towards the end here to kind of give you a little bit of a view of how the types of things we've done. So probably a lot of you have heard of CRISPR. This is an animation that basically shows how CRISPR works. It recognizes, this is Cas9 protein, recognizes a segment of DNA in a sequence-specific way and can cut that DNA. This is being used for genetic therapies now. This is another animation that's one of my favorites that shows how muscle works. And so every time you contract those muscles, you have all of these tiny little proteins that are working really hard to kind of pull these different filaments together, and I'll let them basically come apart. This is why, like doing exercise is so energy intensive, because each of these guys requires a bit of energy in order to kind of pull on those filaments. I've also created animations that aren't molecular scale. I'll play this one because it's pretty fun.
Dust bunnies are complex and include matter from both inside and outside the home, such as hair and skin from people and pets, insect remains, fiber from clothes or furniture, and bits of dirt. Dust is also home to a wide variety of microbes, like bacteria, fungi, and viruses, that come from both human and non-human sources. These microbial communities are surrounded by lots of different chemicals. These chemicals include antimicrobials in our soaps, plasticizers in our bottles, and flame retardants in our furniture and building materials.
Yeah, so it's a molecular animation about dust. Like making this dust bunny was really fun. Like it was not like something I had done before, so I really enjoyed that process. And of course, one of the great things is also kind of going back. So if you went to my talk yesterday, you know that I'm an actin biologist by training. It was really exciting for me to be able to, years after I graduated, to go back and be able to start animating some of the things that I worked on in graduate school in collaboration with my PhD advisor. And so this is an example of that. And in general, I think, these collaborations with researchers, getting to be able to visualize the things that they've studied for years or decades is incredibly rewarding. Like we've had researchers who are almost in tears as a result of being able to have an animation that really kind of depicts their research in the way that they see it inside of their heads. So, one of the things that I think about sometimes, I have friends who are astronomers, and I think about how, jealous I am of people who are astronomers. Because, in terms of reaching the public, getting people excited about what you do, astronomers are so good at that. You don't have to be an astronomer to get excited about images like this, right? So this probably dates me, but there was a time when Apple just by default had these sorts of images as your background images on your laptops, as well as your iPhones. And I think this is, is sort of indicative of their merit, both as a beautiful image as well as, their status as scientific data. But of course, when we think about biology, we do have a lot of beautiful images. So this is a microscopy image of a rat brain that shows, fluorescently labeled cells that you can see in all of these different structures. So David Goodsell, who I mentioned before, makes beautiful illustrations of these molecular landscapes. This is a cell known as Mycoplasma mycoides. And of course, there are people like me who are creating these animations of how we think the dynamics inside cells look. Which I hope you would also find, beautiful as well as scientifically instructive. So, this is my last slide, and the thing that I kind of want to leave you with, and I want to mention, because it's sort of timely right now, is that pretty much everything I've shown you today was funded directly or indirectly by federal funds. So my own group, the Animation Lab, as well as the GSLC, rely almost entirely on federal grants from the NIH and NSF to continue our work in visualization and science education. So, I've encountered many challenges in my career, a very non-traditional kind of career. But I think has been no challenge that has been quite as large as the one I think we're facing today. And so hopefully, these animations show you that there's incredible work being done on the molecular side. And this is just a tiny slice of kind of the amazing science that happens in this country. So I think, the kind of message here is, take some time to learn about what's happening out there right now. There hasn't been a more important time to kind of have your voice be heard if you feel comfortable doing so, and to support science. Yeah, thank you, and I'm happy to take questions. Is this like a question taking this? Is there a Q&A? Yeah, I'm happy to take any questions if there are any. Yes. Oh, is this her?
I'm curious about the marrying of two probably passions that you have, and how does that come about, and sort of how do you navigate that when you came into an academic environment, which tends to be very department focused?
Yeah, so I think, when it comes to grant funding, you can't, I get a lot of research-based grants, and so we have to make the argument that this is important for scientific research, and also make the argument that communication is central to the scientific, kind of endeavor. Researchers need to be able to communicate ideas to each other in order to really, move to the next kind of phase. So I think there's been, you can't really talk too much about aesthetics or about art and those types of grants. But in reality, when we're thinking about doing outreach, it's important for things to look attractive. But in a lot of ways, the kind of the aesthetics and look of things comes out from the science itself. Like the complexity of biology is in itself beautiful, without having to really add much to it at all. And so I think in general, I've shied away from, there are a lot of people who might consider me an artist, and that's not a label that I've really adopted very strongly, partially because I have to, you need an impartiality, I guess, in order to kind of do what I do. So there is a little bit of this disconnect between saying that these things are beautiful, but they're also scientifically valid. But there's historically a lot of examples of people having, had both of those things be true. And so that's what I'm kind of hoping kind of speaks for itself in the work.
I just want to say, it's all super cool, and it's also you couldn't ask AI what happens. It's just nice to know that at the cutting edge of things, you can't ask AI what does this look like?
Right.
So that's just kind of nice, and I just want to know, what do you think is the most if you had an unlimited budget, what would you think is the most pressing, most important thing to communicate or animate?
Yeah, so I think one of the things that I feel like maybe part of the issues today is that science has been hard to communicate. Molecular biosciences particularly is hard to communicate because it's happening at these small scales. Biologists don't feel well equipped in general to be able to communicate the types of things that they study. And I think that's hurt us. And so one of the projects that we're currently working on is trying to create animation software that would make it easier for everyone to be able to create animations. And I think it really kind of gets to the kinds of things that we're studying. We can really kind of, the way I think about the animations is that they're a movie that plays in a scientist's head. So when they're thinking about their research, they have this movie that's based on all of their data that they've collected, that other groups have collected, that forms this sort of like a video of what they've studied, they've spent decades understanding. And the animation is really just trying to get that into a format that we can show the world, basically what people think about. How do viruses work? How do these different mechanisms work that are central for life? But so I think for us, it's really about how do we get to that next level of trying to get biologists to really communicate better, so people can kind of see the complexity, the sort of the different ideas, and also help push science forward. So that's something that we're embarking on now, and that, yeah, hopefully will become a bigger project.
Hi. I really enjoyed both of your talks, and I was just wondering what you think about like the timeline. I know you're building that tool that you talked about yesterday.
Yeah.
To make it easier for scientists to like animate stuff on their own. But do you know how long that would be before that's like rolled out or like implemented widely, or even like implemented in like the medical field?
Yeah, so we've already have like a first version that we're going to start testing internally in the lab, probably in the next month or so. It's right now it's pretty basic. So I think, like we're planning on adding more bells and whistles, but the short answer is like the earliest version of the tools will probably be available later this summer. And they'll be available in Blender. So this is a free and open source animation software. We're going to release tutorials as well. So hopefully, like this is year one of a 5-year grant. So we're hoping to try to, get as many things in there as possible. And then kind of down the road, we're thinking about simulation. So I think we have a lot of ideas of how to make this super useful for people. And also, testing it on different groups like graduate students and making sure that it's as usable as possible. But yeah, short answer is we're hoping, later this year will be the first version, the very kind of the most basic part of the tools.
Hi, back here.
Oh, hi.
Yeah, so something I'm wondering is like, how do you encourage yourself and people that are interested in molecular animation to keep pursuing stuff? Because inherently, as a fourth year bio major, biology can be kind of boring, drawing 2D dimensions. Sorry to all the professors that are here. But yeah, how do you like pursue like being creative and being outside the box and just like seeing biology in like a beautiful light rather than like a 2-dimensional circles and blobs?
Right. Yeah. So for me, I think this has been a passion for a long time. So when I was in grad school, I probably thought similarly that, like even though I was a PhD student in biology, so I think in my mind, biology was super interesting, but we didn't do a good job. Like, all these circles and squares and arrows, 2D stuff, what made science look boring, made biology look boring. And so the first animation that I saw made me realize it's not only about making it interesting for other people, it's also about me being able to understand it. And so I think that realization made me very obsessed with doing animation. And I would say it was really like this total obsession. Like it's the only thing I wanted to do. I was going to figure out a way to do it any way I could. And so I think that's what's driven me is that, and I've gotten, in the beginning, I would say it's hard. It was hard because there weren't people in the field, especially not PhD scientists doing animation. But over the years, I've gotten more and more validation that this is worthwhile and this is interesting, both from scientists as well as from other people. So I think that took a while. And in the beginning, it was purely my own obsession that was driving me forward. And now it really is like, this process of collaboration. People have realized that this process of creating an animation gives you new ideas. People have come up with new hypotheses. We publish a lot of scientific papers. So I think it's really, but in the beginning it was hard. Like you just had to kind of like nose to the grindstone, like blinders, people being discouraging and just like ignoring them. That was a lot of like the early days, to be honest. So yeah, for sure. I think it was just pure obsession that kept me going then.
This is kind of similar to the last question.
Sure.
But I know you mentioned the importance of being surrounded by people who are passionate about their goals and like being driven. So I guess my question is, who was that person for you, like when you first started, and like how did they motivate you to like keep going and like keeping passionate about your interests?
Yeah, I think for me, a lot of it was more internal. Like I wanted to do something, but I didn't know if I could. So I think in the beginning in grad school, I was basically given permission. Like I was basically told, you can do this, you can spend time doing this by my advisor. And so that really allowed me to explore a path that I don't think I would have explored. So over the years, I've had a lot of, so I mentioned in yesterday's talk, I had a program officer at NSF who supported me for a postdoctoral fellowship. So I think the one thing that I've always done since grad school, and partially because my advisor was so supportive of me doing animation, very unconventional thing as a grad student, was that I was always kind of not, I wouldn't say loud, but I was always vocal about saying, this is what I want to do, and this is why I think it's important. And some people will be like, disagree, and some people will be like, I'll help you. And so I think there were enough people who were basically in that latter part that said, I agree and I'll help you kind of make that happen. And so I think for me, there was a lot of people who were in that category, including people at the University of Utah who gave me a tenure track job, despite that not being a kind of a normal thing for tenure track faculty to do. And sometimes it takes time. Like sometimes people in the beginning will be like, I don't know. But then over time, they're like, you know what? You've convinced me. And so I think a lot of it is about perseverance and just never kind of trying to see the other side. Like I can see why this person might not believe, see things exactly the way I do, but I'm going to convince them. And being kind of like nice about it, right? I understood when people didn't quite see it. And you're patient, over time they might see it, they might not, but then you find other people to kind of help you along that path. So yeah, so no one person, but like an entire community has definitely helped.
Thank you. What a fascinating talk. I have a question. As AI is moving forward and we are experiencing concerns about guardrails around AI, do you have worries about how AI and things like hallucinations or inaccuracies may impact your work? And could you describe an example?
Yeah, I guess it's less to do with AI, to be honest, because if you ask AI to create like a scientific animation, it's so far removed from anything that looks like anything that I think it would be hard to convince anyone that that's actually any sort of reality. I think the things I worry about more is like people intentionally just creating animations that are wrong and misleading. And I think that has been the case. So I've been asked to create animations that are clearly total bunk, and I've been like, no, that's not based on any sort of reality. I refuse. But not, I'm trained as a scientist. I would ask for, the publication, show me that this is the way it works. But not everybody would. And so, with animation, you can animate anything you want, including things that are total kind of like nonsense. And that worries me more. Like, of course, there are museums where like people are walking around with dinosaurs, things like that you can animate and it looks pretty real. For some, it might be convincing. So I think that's what I worry about more, is less AI than just like people, people, and, eventually AI as well. But I think, sometimes it's, people want to mislead. Yeah.
I have a question up here. Sorry.
Oh, hi, yeah.
Hi. So my question is, how do you balance scientific accuracy with like visual appeal in your animation?
Yeah, so we do take a lot of shortcuts. So some things that, I talk to people about is like, we don't show crowdedness. So cells are crowded with proteins. They're just packed full of proteins. And we don't show that most of the time because it would be distracting. So, a lot of times we have like something that looks like a cellular vacuum, and like, the protein we're interested in is shown in the forefront. So I think we do make, kind of like, we don't show a lot of what's happening. Also, like, our time scales are often exaggerated. Molecular events happen over the course of often milliseconds to seconds, sometimes minutes, and we will just do whatever kind of makes sense in our animations. And so there are a lot of those kind of things that aren't really shown completely accurately in the animations. So yeah, so sometimes we definitely, are trying to tell a story and make some concessions for that.
Well, thank you everyone, especially for those who stayed for all the questions. Thank you so much, Janet, for coming here. Thank you, Matt and Ben. I'm not sure how I got chosen to pick the speaker, but honestly, it's like this is a once in a lifetime thing. So thank you so much. I forgot, some students and I got you a blanket because it's so cold outside that says Augsburg on it, but I'll have to give that to you at a later time. And Dr. Iwasa will be at the poster session this afternoon, so you can interact with her there as well. All right, thank you again.
Thank you.